Hi, Miss Nikki here. Welcome to Chapter 5. We're going to do a quick little summary on the most difficult topics of cell division. We have to talk about two processes, mitosis and meiosis. But first, we're going to talk about the cell cycle and kind of lead into mitosis. So cell division is how we take one fertilized egg, that means there's sperm there too, so sperm and egg, and grow into an organism with trillions of cells. So we start from one cell and we're going to grow into our trillions, and we do this by mitosis. So if you see the term somatic, somatic is talking about all the body cells that continue to undergo cell division even as an adult. So we're constantly making new linings to our esophagus, to our intestines, to our cheeks, right? So thousands of new blood cells, skin cells, anything that lines the respiratory tract. We do have to mention apoptosis. This is programmed cell death. Most of us think of lysosomes when we think about apoptosis. So if we need to decrease the number of cells, or if we want to get rid of any unwanted tissue. So they talk about the webbing between the fingers and toes. They talk about the tail of a tadpole, but also think about just taking the trash out. So what if you have an organelle, like one of your mitochondria or Golgi apparatus, or you have a vesicle, something is not functioning the way that it's supposed to, how do you get rid of it? Well, we get rid of it through apoptosis or programmed cell death. Here we're showing the different stages of the cell cycle. So this whole thing is the cell cycle. You're going to see a G1 stage, which is growth. You're going to see a G0 stage. This is basically when cells are kind of outside of the growth phase, if you want to call it hibernation. Basically, these are cells that are not growing. Couldn't spell there for a second. So these are brain cells, heart cells. These would be in G0. But if you have a cell that wants to grow, so you have one cell and we need to copy it and make a new cell, it's going to go through G1, which is growth. Then it's going to go through the S phase. This is when the DNA is going to copy itself. Then we go through a secondary growth phase for basically making the final preparations for division. And then all of this here is mitosis. So we do a lot of prep work. So the cell spends the majority of its time in interphase, which is all of the prep work for mitosis. I like this image because it's showing you what we mean by replication. So here is a chromosome, right? And this chromosome has to copy itself. Because what's going to happen? You start with one cell and you're going to end up with two cells. So this and this have to be able to be pulled apart. This is happening in interphase, but specifically S phase. So you may have noticed before these little checkpoints. So the cell cycle has checkpoints to make sure that the DNA is not damaged and that everything is going according to plan. So you have this first growth phase, and then we have a checkpoint, right? Is the DNA damaged? Is the cell growing appropriately? Do we have all the nutrients and everything needed to move to the next phase? Then the DNA gets copied. And then we grow a little bit more and we have this final checkpoint, this G2 checkpoint. This is where we really want to make sure that all of the DNA copying was accurate, right? So the DNA has replicated properly. Apoptosis will occur if the DNA is damaged. We do not want to copy a cell if the instruction codes are already wrong, right? Then instead of one bad cell, now we've got two. So before we enter mitosis and pull those two chromosomes apart, we're going to do another checkpoint here in the middle, and this is when they all line up. We'll go through each of these phases, but we do midway through mitosis. Is everything going okay? Do we need to finish production? So two ways that the cell cycle can be altered are proto-oncogenes and tumor suppressor genes. So this one here in green, this is if we increase stimulation, and this one is if we decrease. So what does this mean? Everybody has proto-oncogenes. They code for growth factors. Basically, these proto-oncogenes tell the cell to divide. Cell, divide. But what happens if this DNA segment is altered? So if the DNA is altered, your proto-oncogene can turn into what we would now call an oncogene, and basically it's telling your cell to divide all the time. It's sending these signaling proteins to tell the cell divide, 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 divide uncontrollably, and that can produce a tumor, right? And then if that tumor invades other tissue, then we call that cancer, right? So a tumor suppressor gene is the opposite. That tells you what they do. They suppress tumors. So you have a normal section of your genome that's an inhibitory pathway, and it basically inhibits cell cycle. So you kind of have this yin and this yang. Turn on the cell cycle, turn it off. Turn it on, turn it off. If your proto-oncogene changes, now your signal is on all the time. You're copying. Well, what happens if your tumor suppressor gene gets turned off? So this tumor suppressor gene gets damaged in some way, shape, or form due to a chemical binding it, maybe just a random mutation in your tumor suppressor gene, and all of a sudden you have no more inhibit. So instead of inhibiting, you're telling the cell to divide. So we could have two areas that are supposed to be doing the opposite. Now they're both driving the cell to copy itself repeatedly. So everything before, we were kind of talking about worst case scenario. Now we're just going into the overview of mitosis and meiosis. So what should we normally do? We should have a cell, and that cell should replicate its DNA. It should also grow, and then that cell should split into two cells, and these should be identical. So you start with this cell that has 4 chromosomes, and you end up with 2 cells that each have copies of the same chromosomes you started with. So they're identical. Of course, there's a little more to it. I'd like you to look through each of these phases and make sure you can put a sentence together about what's happening. So at first, we're going to see these centrioles become active. They start moving towards the poles, and you'll see the DNA goes from this string-like chromatin to where you can actually see the chromosomes. And what's going to happen is they're going to line up, once the centrioles move to the opposite poles, these are going to line up in the middle. We can see here in metaphase that the chromosomes are lined up in the middle. What's going to happen next is anaphase, where they get pulled by the microtubules, or it's sometimes called spindle apparatus, pulled away to the opposite sides of the cell. And then we have this cleavage furrow, and you can see here in the microscope image. And eventually telophase, these will separate and you'll end up with 2 cells. There's only a few things different in plant cells. So you're going to see that the chromatin is condensing into chromosomes, that the nuclear envelope is breaking down just like before. But here we're going to see centrosomes. You're not going to see spindle or centrioles. You won't see those in plants. So in prophase, now we see more chromosomes. In prometaphase, we see them kind of migrating towards the middle. Metaphase, they're lined up. Anaphase, they get pulled to the side. Telophase, we start to see daughter cells form. I want you to notice this word, cell plate. So remember how in animal cells they called it a cleavage furrow, it kind of went from the outside in. This is a cell plate. You're basically building a wall, a cell wall between these 2 cells. So that's something that's a little bit different in plants and animals. Now we're going to talk about meiosis. So meiosis only in ovaries and testes. Basically, meiosis is where we half the chromosome number. Remember, mitosis is identical. If you have 4 chromosomes, you end up with 2 cells with 4 chromosomes. And remember, mitosis is all body cells, somatic, that term, right? So what do you notice here? You start with 4. They replicate, copy themselves. They split into 2 cells, and then those cells split again. You started with 4 chromosomes. You end up with 4 cells that have 2 chromosomes. So did we halve the number of chromosomes? Yes, hopefully, right? Okay, here's the first division. So we start with this chromosome in blue and this chromosome in red. They're both, let's say, chromosome number 1. You got the blue one from your dad, and we're going to say you got the red one from your mom. Now you have one of each, right? You've got one chromosome from mom, one chromosome from dad, so you have 2 number 1 chromosomes. That is going to undergo DNA replication. We're going to copy all of the chromosomes. So now do you see you have a copy of dad's, you have a copy of your mom's, and then a copy of your dad's number 2 and a copy of your mom's number 2. So you have just doubled all of your chromosomes. Look at how they line up in metaphase. I say they line up like shoes. They look like pairs of shoes, right? So these are both chromosome number 2, and these are both chromosome number 1. You've just made extra copies of them. We still see the centrioles. We know we're doing division. They're still lined up in the middle. You also see how little bits have swapped, right? So during prophase, some of the chromosomes that are laying on top of each other are going to swap spaces. I'll show you why in just a second, or what it leads to. Then we have the same process. We have anaphase where we drag them to the poles, and then we have our daughter cells that are created, and we end up with 2 cells. Now, if you look at these, you can see they're not exactly what we started with, right? They've got little bits that are swapped, so they're not exactly the same. So this is the first division. Sometimes you'll see it referred to as Meiosis I. And then we're going to move into Meiosis II, the second division. Here's a nice image showing you the crossing over. So this is your original chromosome 1 that you inherited from your dad, and here's your copy. And then here is your mom chromosome number 1 that you inherited, and here's your copy. Remember, we did duplication. So this crossing over can occur, and you will actually swap pieces of DNA. What we're going to see, remember how we said we start with 1 cell, and we end up with 4 cells that have half the chromosomes. So each one of these is going to end up being a cell. So 1 cell, 1 cell, 1 cell, 1 cell. These are going to end up either being egg or sperm. They have half the chromosomes that you started with. This is why you could be tall and have blue eyes, just like your dad. You could be tall like your dad, but you have green eyes like your mom. Maybe your green-eyed allele is right there. Then this one here, you're going to be short like your mom. But guess what? You got blue eyes like your dad because the gene's right there. And then this one, you're going to be short and have green eyes just like your mom. So this is how we get variation with our offspring because of this crossing over event that happens during prophase 1 of Meiosis I. Now we move on to our second division. So they're just showing you the whole process. Here we have these 2 cells. Remember what came above here. This was the first mitosis division. Now the cell is going to go through the same process again, but do you notice the numbers at the end, the Roman numerals? So there's anaphase 1 and then there's anaphase 2. You need to know what's happening in each of those, either in Meiosis I or Meiosis II. It really only changes for some of them. So like this one's important, metaphase looks a little bit different, and prophase is a little bit different. The rest of them are similar. So what do you notice this time in metaphase 2? You see how the chromosomes line up in the middle exactly like they did in mitosis. Remember before we had what I called shoes, right? They ended up like this, and then these guys went this way and those guys went that way. All right, so now we have a little bit different lineup, but it looks really similar to mitosis. Let's do a little comparison. Here's meiosis, here's mitosis. So in meiosis, we start prophase 1. Remember the important event that happens, the crossing over that occurs. Does that happen in prophase of mitosis? No. We want identical, right? What do we want in meiosis? We want variation. So two totally different goals. So now we can see our crossover event occurred. In metaphase 1, the homologs. So these are homologs. Are all four of these chromosome 2. Yes, you have 4 copies of chromosome 2. That's why they're called homologs. All right, in mitosis, we have this metaphase where everybody just lines up in the middle because they're going to be split right down the middle, and that chromatid is going to go that way, and that chromatid is going to go the other way. In this one, what's going to happen? Do you split chromatids? No. What do you split? Homologs. So that's a big difference between metaphase 1 of meiosis and metaphase of mitosis. So now we go through anaphase and telophase. We go through the second, so meiosis 2 would happen after this, right? So that would go into meiosis 2. We go through the metaphase, anaphase, telophase again, and we end up with 4 daughter cells that are not genetically identical. Remember, this one's got that blue little piece, and this one's got a red little piece. All 4 of these are different. They're not the same as the cell you started with. These guys, these daughter cells with mitosis, they are genetically identical to the parent cell. So think about it for a second. If you're trying to copy your skin because you have a cut, you want your skin cells to make more skin cells. You don't want them to be different. Meiosis, what's our goal? We're trying to make egg and sperm. We want different. We want variation. It gives us a better chance at survival. These are some good slides in the book if you want to go through each phase and give kind of a basic definition of what's happening, how they're similar. So I would try to do some comparison of the two. There will be a few test questions about why one is different or in which phase does this happen compared to mitosis when this happens. So this is the cycle of life in humans. So you have dad who's making sperm, and you have mom who's making eggs, right? And what did we do? This was meiosis because it's ovaries and testes. So we went from, I'll use us, humans. We have 46 chromosomes. We went from a cell that had 46 and we split it into 4 cells that have 23 chromosomes each. Did we half the number of chromosomes? Yes. Why do we do this? If you have an egg that has 23 and you have a sperm that has 23, what happens when we add them together? We have 46 chromosomes. That's what this is. This is a zygote. It's when the egg and the sperm come together. You now have recombined your 46 chromosomes, but remember they're genetically different. Again, this gives us a better chance to adapt and survive in a changing environment. But then we have to grow from this zygote, this one cell, we have to grow into a baby, and then we have to grow into adulthood. So this is where we're undergoing mitosis. I wanted to show you a quick difference between spermatogenesis and oogenesis. So this is still meiosis. We're still talking about meiosis. We're still talking about 2 divisions. But I want you to see that you start with one cell that has 46 chromosomes, and you end up with 4 cells that have 23 chromosomes. And you create 4 sperm. With eggs, it's a little bit different. The egg has to be larger. It has to carry all the nutrients for the growing zygote until it can implant in the uterus. So what happens is this cell is still dividing, but you get one giant egg, and then you have something called polar bodies. So I'm going to draw little polar bodies over here. So this process is still happening. You still have meiosis division 1 and 2, but basically the polar bodies are not going to be fertilized. They're small. All they have in them are chromosomes. The egg grows giant, filled with nutrients. So really, when you start off with a cell in the ovary that's got 46 chromosomes, you're really only going to end up with one giant egg that has 23 chromosomes. The process is still the same. We just kick out the other small little cells and we call them polar bodies. Here's the second half of that comparison table. Again, just go through these. Make sure you know mitosis and meiosis. You know the difference between meiosis 1 and 2, and then you go through each phase and tell me something about it, and then compare the two, how they're similar, how they're different. If you have any questions, please let me know. Bye.